Stephanie, do you know how to admit students?
I do not.
Okay, I will try to keep an eye on that then. Actually, it'll just come up and warn me.
Okay.
All right, so we're going to start with the heart. That's where we started in this unit and went all the way through the immune system. With the heart, one of the things you need to know is the anatomy. Don't worry so much about that first history kind of lecture where we talked about fish and how we developed into these four-chambered hearts. Just recognize that we now have four-chambered hearts. They're essentially two pumps working in concert in order to be able to get our blood oxygenated and then get that oxygenated blood out to the rest of the body. So this is a picture that Callie Clements, one of our UTAs, had drawn. I stole it from her because even if I took my time, I could never draw it this well. This is an image of the heart if you were to cut it open and then split it out so that you could see the chambers inside. So we've got several different layers of this that you should be familiar with as well. We've got these mesothelial layers. Mesothelium night.
Hey, Michelle, your sound is cutting in and out. I can't hear anything from you anymore, and I'm getting that in the chat as well.
Okay. Well, this might not have worked very well. I'm going to try going back to how about now?
I could hear that.
Yeah, so it would help to be plugged in.
Yeah, that sounds good.
Good. Thank you for letting me know. I'm also having some problems with my pen. So it's a good thing I pre-drew a lot of this. Mesothelium. So mesothelium is the pericardium around the heart. It's the two layers that have fluid in between them, and they're going to keep the organ in place, and they're going to prevent some friction from happening. So you always have these two layers. The outside layer is going to be the parietal layer. The inside is always going to be visceral.
Bye. Bye.
I always remember that visceral means gut, so it's always the one that's on the organ, that's the visceral layer, and then the one that's connected, in this case, to the chest wall, the chest cavity, thoracic cavity, is going to be the parietal pericardium. From there, you actually get into the myocytes, the muscle cells. So this is what you see when you look at the outside. It's what's typically drawn as red. Okay. So we'll just put that here in red then. So this is myocardium. Myo means muscle, cardium means heart. And we've actually drawn in here some of those sarcomeres. These are going to be contractile cells that have those sarcomeres, the Z lines with the actin and myosin, and they're capable of contraction. Underneath that, you actually have your chambers. And so you're going to have, we'll say that this is the right side of the heart, and this is the left. So this is going to be the right atrium. And in here is the left atrium. The bottom parts are the right and left ventricles. And then surrounding this is going to be your endocardium. Remember that anytime we're talking about a vessel that has blood in it, it's not an epithelial cell, we call it an endothelial cell. But here in the heart, we have a special name, and that's the endocardium. The other thing you want to make sure you are comfortable with is all of your valves. So you've got valves that are here in between the right atrium and ventricle. This is the AV valve. So named because it goes from the atrium to the ventricle. This opens in. Both of these are going to open in from the atrium to the ventricle. So when you bring blood back and you put it into the atrium, it can just push that valve open. That's like a door that, as long as there's not too much pressure built up behind it, you can push it open and the blood will passively pour into the ventricles. Once you start contracting your ventricles and you build up pressure behind that door, it's going to slam it shut. And then you can't get any more blood from the atrium into the ventricle until you relax again. Once you start that contraction and you slam the AV valve shut, you can start building up pressure because that blood has nowhere to go, but it's being squeezed into kind of a smaller space now. And so you have these valves that are going from the ventricle out to a blood vessel. In this case, here on the right side, this is going to be a pulmonary valve because it's going into the pulmonary artery. And then on the left side, when you push this out, it's hard to see, but it's going this way. So I'm just going to mark this as 1. That is your aortic valve because it's pushing into the aorta. By the way, these are the vena cava that are coming into the atrium. You've got superior and inferior. And then when it goes out through those pulmonary arteries. It will come back into the left atrium in pulmonary veins. So this is a good example of a time when your terminology for arteries and veins are not going to be, it's not going to be in line with whether or not it's oxygenated. So it's always going to be artery moves away from the heart, veins move toward. And in this case, from your right side of the heart, you're bringing blood back that's already deoxygenated and sending it to the lungs. So it's going through the pulmonary artery, but it's deoxygenated. And we can kind of see that better on the 4-box model. So again, this is going to be right atrium, left, and your ventricles. This is going to be, let me move this. Nope. Blood coming back from your body. It's deoxygenated. It's going to go into the right atrium. It's going to go through this right AV valve. And then from there, it can move out the pulmonary artery. And the pulmonary valve. It's going to go to the lungs, it'll pick up oxygen, and then it can come back through these pulmonary veins. So these are now going to be oxygenated. This blood can then pass through the left AV valve. Into the left ventricle, and then from there it can go out the aorta. But again, in order to be able to get out either the pulmonary or the aorta, you have to be able to build up enough pressure so that there's more pressure on this side to be able to swing that door open and push it out. Your right side of the heart, since it's just going to the lungs, it doesn't need a whole lot of pressure. It is able to push through this pretty easily because it's just pumping to the lungs and then back again. But when you're talking about the left side, you're talking about trying to get it all the way to the heart and then all the way down to the feet. And in order to build up pressure enough to do that, it's a lot more pressure to have. And so you're going to carry a lot more pressure into the aorta and through your system. Okay. You should know your TOAST diagram. This is TOAST here, also known as the pressure volume curve. TOAST is cute. I like TOAST, but pressure volume curve tells you what it is. So it's pressure on the y-axis and volume on the left axis. And that tells you a lot that you need to know about this. So when you see things moving along this, if you've got movement in this direction, it means you're increasing volume. If you're moving in this direction, then it's an increase in pressure. And that tells you what valves are open or closed at any given time. Because to move volume, you have to have a valve open. It has to be able to push through a valve to be able to move. When you're increasing pressure, you cannot have any valves open, because if you start squeezing and there's a valve open, you're just going to shoot the blood out and you'd never be able to build up the pressure. So you know, since we're moving volume here, this has to have a valve open. Okay. And this, because it's so low pressure, this has to be the AV valve. There's no way you could open the pulmonary or the aortic valve here. So this is AV valve opens. And so that's going to be moving blood from the aorta into the, or sorry, from the atrium into the ventricle. And this is called diastolic filling. At the end of your diastolic filling, you're actually going to contract the atrium and squeeze the last remaining blood from the atrium into the ventricle. And so that's why you see this increase of pressure just at the tip here. Once you start moving up on this graph, you know that the valves all have to be closed. So we're just going to say all valves closed. And that's because we've started to contract. And so that AV valve is closed, but has slammed shut, and we haven't built up enough pressure to open the aortic valve. This pressure volume curve can actually be for either the right or left side of the heart, but we typically look at it in terms of the left. So I'm just going to label this as left, and we're going to talk about the aorta. All right, so this portion here is going to be called the isovolumetric, iso meaning same volume, contraction, because we're building up that pressure. Once we get to the top, we have to open a valve to be able to change the volume again. So this is going to be the aortic valve because it's high pressure. Once you push that valve open, now the blood can actually escape into the aorta. I'm doing all these like hand gestures because I forget you can't see me, but this is ejection phase. And you're going to continue contracting, so you'll still see some increase in pressure while you're seeing this decrease in volume, but eventually the pressure is going to get back to the point where the aortic pressure is going to be higher than the pressure in the ventricle, and at that point the aortic valve closes again. And then from there you move into the isovolumetric relaxation phase. The other thing you need to be able to take away from this is the stroke volume. So we say that the stroke volume is the amount of blood that we can push out in one heartbeat. So we've filled up this ventricle and then we've pushed blood out of it. And at the end, we have a final volume. So this is going to be the end systolic volume here. What we actually were able to fill when we put the blood from the atrium into the ventricle is the end diastolic volume. And just a reminder that diastole is during relaxation, so that's your filling phase. Systole is contraction. So it's the point from, I'm going to do this in green. So this was systolic volume. This is systole here, the entire contraction phase. If you subtract the two of them, if you take EDV from ESV, or the other way around, if you take ESV from EDV, then that gives you that total volume that you were actually able to move during that heartbeat. So from that, you can also calculate something called cardiac output, which is the total amount of blood that you're able to put out per minute. And that's going to be stroke volume, which is how many or how much volume you were able to put out per heartbeat. And you multiply that by how many heartbeats you have in a minute. So that's your heart rate. And that's you get your cardiac output from that. Finally, what I want to show you is that you can see this ECG with ion movement, and you can overlay that onto the pressure volume curve so that you can see where these things are happening. So this is just going to tell you what ions are moving, not necessarily what direction. So I know, it throws people sometimes that like the potassium efflux has a positive deflection, but this is based on running from a lead to a lead, and depending on what direction you're looking at. So positive deflection just means that it's running towards you, towards the lead that you're reading, and a negative deflection just means it's running the opposite direction. So it depends on where the leads are. But this is going to be your P wave. And a P wave is measuring sodium influx during atrial contraction. So we know we could see the atrial contraction here at this little increase in pressure, and that's your P wave. This green is the QRS complex. And honestly, there's a lot here in the QRS complex, more than we get into. It's fascinating to really kind of look into an ECG and all the things it can tell you. But for what you guys need to know, we just expect you to understand that this is also sodium influx. But at this point, we're looking at the ventricle contraction. In other words, this is systole. Okay, so this was the screen. That's QRS complex. And then finally, we have this T wave. And T wave is going to be potassium efflux. So that's your repolarization. That is your relaxation phase. Relaxation. So that's this section, isovolumetric relaxation. Okay. So that's kind of talking about systemic circulation, but we also have to feed the heart because the heart muscles need oxygen to be able to function and contract and be able to push the blood to the rest of the body. And so we have this coronary circulation system. Coronary circulation is coming off of the aorta. So this is the aorta here. And you've got these coronary arteries. I don't know if this is going to show up well. Yeah, I guess. These are actually feeding out of the aorta. This is above the aortic valve. So that's, we'll say, the aortic valve. The problem is when we build up so much pressure and we're shooting it out the aorta, we're doing that because we actually contracted the heart to build that pressure up. And these vessels are ingrained into this muscle. It's actually built into it. So when you contract the heart, you also squish these coronary arteries and all the veins and venules. And so you can't really push blood through. So the only time you could push blood through is once the pressure drops enough that this aortic valve closes again and you're in systole. So this can only happen in, you're in diastole, not systole. You're in that relaxation phase. What happens is you're pushing this blood out But as the pressure drops, you may not actually be able to get some of the blood over that hump. So it's going to fall back down. But because you just slam that aortic valve shut, it's going to pull down here. These coronary arteries are sitting right here. And now that the heart's relaxed and they're no longer squished, this can now just drain down the arteries and feed the heart. Okay, I know no one really likes math, but there's a couple of equations that you should know. You should be familiar with Ohm's law, which tells you that the change in pressure, which we consider to be constant, because anytime you're looking at the difference in pressure from, say, one particular artery to an arterial, is not going to change. That's going to be equal to flow times resistance. When we're talking about blood and we talk about resistance, we're talking about the friction that the fluid is going to experience against the walls of the vessel. So when you push all this blood out, if you have a nice big vessel like the aorta, there's not going to be a whole lot of fluid in contact with the wall compared to the amount that's actually able to flow through the center. So the stuff that's on the wall will have that friction, but the stuff that's moving through the center has no resistance. When you have much smaller vessels, you've got more of that fluid that's in contact with the wall, and so you increase resistance. When you increase that resistance and you have more drag on the fluid, it makes sense that it would slow down the flow. So these are inversely proportional. If you increase your resistance, you decrease flow. Okay, the other one you want to know is Pascal's law. And that's, you know, one of those really long, complicated formulas that we can just get rid of a whole bunch of things, and it breaks down to flow is approximately equal to the radius to the fourth power. So it's telling you that anytime you make a change to the radius of these vessels, you're going to have a factor 4 effect on flow. So if you double the radius, you increase flow by 16, 2 to the fourth power. But I also want to point out that if we now are saying that flow is approximately equivalent to r to the fourth power, you could actually replace that here and make this r to the fourth power. And so now you can see, although we kind of knew that intuitively, that if you decrease radius, you increase resistance because you're going to have more of that fluid in contact with the wall, and vice versa. These principles are really important in coronary circulation because if you change that radius, you can do that by modulating the smooth muscle around it, but you can also change the radius by building up plaque inside your vessels. And that's not as big of a deal if you're doing it in something as big as the aorta, but when you start building up plaque in these tiny little coronary arteries, you're going to have a significant impact on that flow. And because this is feeding your heart, and you need these heart muscles to actually get the oxygen and be able to contract, if you block that flow, if you decrease it too much, you're going to have a heart attack. You're not going to get the oxygen that you need to the heart muscles, that tissue will die, and then it will no longer be able to contract. You do enough damage, and you're not going to be able to pump the blood out to the rest of your body, and that becomes a big problem. All right, so let's talk about how we actually move this signal. We actually get our heart to contract in the first place. So this is going to be the conductile system, or I'll just say the conduction system. I think I was very optimistic about how much space I was going to need for this stuff. So this is my version of the heart when I'm not using Callie's drawing. This is going to be the right atrium, left, and your ventricles. So in your right atrium, you're going to have 2 pacemakers. First one is going to be a bunch of pacemaker cells together in the SA node, sinoatrial node. This is actually going to feed the atrium. So this will send signals to both sides of the atrium for contraction, but it's also going to feed to the AV node. And that's named because it's actually, I don't want to write that there. Because it's on the septum between the atrium and the ventricle. So these nodes, these pacemaker cells, are capable of creating a rate and rhythm on their own. They don't need input from the brain to tell them to beat. So SA and AV nodes. Once they start that rhythm, they can pass that now through these bundles. These are going to be conductile systems that are going to run along the septum and then split out between the two ventricles. And then from there, feed into every part, all the myocardium that's on these ventricles. Okay, so this first part here, I'm going to label 1, 2, and these are 3. 1 is going to be bundle of His. Then when it splits, we call it left and right bundle branches. And then 3 is going to be Purkinje fibers. We call these either conductile or sometimes transition cells. Recognize what they are, like that they're the bundles, and recognize that they're feeding this potential from the pacemakers to the contractile cells, but don't worry about like the channels that are on them or the potential, what it would look like. We're just going to focus on the pacemaker and the actual contractile cells. The conductile cells are kind of complicated, so we kind of ignore them. And then finally, when you're actually feeding each one of those myocardial cells, those are the contractile cells. Okay, so this is all a network. They're very much connected. You start with this pacemaker cell, and you're going to have a special channel on here called an HCN channel. That's the hallmark of these pacemaker cells. These HCN channels are sodium channels, but they function by not only voltage, so a hyperpolarization could set them off, but they also act as like ligand gated channels because they need CAMP to bind to it to open. So you've got to have both a low voltage in here. So if you have lower voltage, and you have this CAMP that binds, and it can bind to 4 sites. Then that's going to let the sodium in. And we know that those sodium is then going to help to polarize the cell. And we'll look at the actual potential for that and walk through it, because we also have a calcium channel in there that will do the full depolarization. So we'll draw in a calcium channel. This is going to be a voltage gated calcium channel. So it will open once there's enough positive charge coming in from sodium. It's going to let that calcium in. That brings in a whole bunch more positive charge. That positive charge can now actually pass to the conductile cells because they're connected. They're not only locked into each other, okay, so it has these junctions here. It's also going to have these gap junctions, these connexons in between them. So you'll have channels that pass from one to the other. So that positive charge that you bring in for both the sodium and the calcium can now spread over to this next cell. And again, we're totally going to gloss over what happens with the conductile cell, the channels that are happening, and we're just going to say that there are channels here. It's going to cause a potential. It's going to bring in some more positive charge, And because these are also connected by these gap junctions, which I really should have labeled, that's going to allow that to pass now to the contractile cells. And this is another one we will actually talk about all the channels on. So here you're going to have voltage gated sodium channels. These are very much like what we've talked about every time before. These are channels that are going to be opened by a depolarization. So positive charge opens them. They're going to fully depolarize to their equilibrium potential, and then that's going to cause the opening of that positive charge. I'm going to draw some up here too. So these are voltage gated sodium. That positive charge will move along the membrane, and it's going to cause the opening of DHP receptors. Here, DHP receptors are voltage gated, meaning that positive charge doesn't just cause it to move, it actually causes a gate to open. And since these are calcium channels, we say that this is a voltage gated calcium channel. This is now going to allow calcium that's in the extracellular space to move into this TSR junction. And on the SR here, we're going to have another channel, and this is the ryanodine receptor. This is a ligand-gated calcium channel. So this calcium that came in through the DHP, because we had the depolarization from the sodium, can now bind to the RYR receptor, and that's going to allow the calcium that was in the SR to move out and into the sarcoplasm. So we know the story. We talked about the story in skeletal muscle that calcium can now bind to the troponin C. Troponin C will then activate and cause tropomyosin to move. That opens up a myosin binding site. The myosin heads can come up and bind to it. The ADP will move out. That's going to cause a power stroke. Then ATP comes in and you drop the myosin heads off. It cleaves to ADP and it resets, and then you could just keep doing that. So it's the same contraction story. It's the same mechanism in terms of the sarcomeres with these Z lines and the myosin and actin filaments. It's just a matter of how we're getting that calcium to those sarcomeres in the first place is a little bit different. There's so many questions in the chat. You keeping up okay?
Yeah, so far. There was one question. Are these notes going to be posted later, filled out?
I don't know.
I wasn't sure what notes they were referring to, but I said that I would ask you.
Okay, yeah, my assumption is that they're actually talking about what I'm drawing here.
Oh, okay.
And I'm not even sure how I would go about doing that, but I mean, you can play the video back and just screenshot the stuff. I'll work on seeing if I could find a way to print it. The last time I tried to print this stuff, it was like 50 pages. So I'm not sure if I can, but I'll look into it. Okay, I'm going to see if I can do something. Oh no, I really screwed myself up. There we go. Oh, you son of a gun. Okay, now that I've given myself a little more room, let's look at the potentials for each of these. So we said we had the pacemaker potentials. And what makes the pacemaker so different is these HCN channels. They have this unusual potential, like they create this different potential where it's going to be a ramp. So you're going to start from rest, and then you get this ramp. So that's from your HCN. Once your HCN reaches threshold for this cell, that's then going to cause, what color did I make calcium? Purple. That causes a full depolarization. Now, this is what we would typically see with the voltage-gated sodium channels, but this cell is just a little bit different. So this is the voltage-gated calcium channel. We'll do the depolarization. And then, oh, I forgot to draw in potassium channels. We got to have potassium channels. You always have to have your potassium to be able to bring it back to repolarize it or hyperpolarize it in this case. So your voltage gated potassium will repolarize it, and then like most voltage gated potassium channels, it will cause a hyperpolarization, which is brilliant because we need hyperpolarization to open these HCN channels. So once we've done this, once we've had these voltage gated potassium channels bring this to a hyperpolarization, as long as there's camp there to bind, the HCN channel is automatically going to open again. So now we have another potential because this will reach threshold. It's going to kick open those voltage gated calcium channels, and then the potassium will hyperpolarize it again, and you just keep going. So this is why your heart can beat every minute of every day for your entire life, because it just keeps repolarizing, hyperpolarizing, and setting off another potential. If we're talking instead about the contractile cells, this looks a little bit more like what we've talked about in terms of neurons and action potentials. This is going to have a resting phase, a depolarization. It does have this kind of funky plateau that we haven't seen. Oh, I should do these different colors. And I should see what color I used. Okay, so for contractile, I did gray for purple and I'll say blue. So we'll keep it the same. So here's rest. Here's our voltage gated voltage gated sodium depolarization. Then we have this voltage gated calcium plateau. And then we repolarize through voltage gated potassium. This does not automatically regenerate, and it doesn't need to because your pacemakers are already creating this constant rate and rhythm that is being passed through those gap junctions to the contractile cells. So there's always going to be the slight depolarization that comes in from this positive charge that passed from the pacemakers to the contractile cells. So you get that little bit of positive charge, it starts to open up the voltage gated sodium, and then you kick them all off. The other thing I would point out here is this, we can name, this is a DHP receptor. Okay, so this is the one that was on the T tubule, and that's going to allow the calcium to move in from the extracellular space. You really can't see the RYR in this potential. Okay, so while we can use the heart to just maintain and create and maintain this rate and rhythm, we can also modulate it through the sympathetic system. Or I should say the autonomic system. We can do either sympathetic or parasympathetic. So sympathetic is going to be fight or flight, and parasympathetic is going to be rest and digest. So this is parasympathetic we're showing here. And this is sympathetic down here. I know that just by looking at the length of the different neurons I've drawn. So with both of these systems, these are descending systems. There's two neurons in a row. You're going to have a pre and a post. So this is your preganglionic, and then a postganglionic. And in the parasympathetic system, you're almost always going to have these really long preganglionic neurons that are going to go almost all the way to the target, and then just a short little postganglionic, and it's the reverse for sympathetic. So this again is pre and post. This is going to have these really short pre and then long postganglionic cells. Excuse me. And those names are telling you kind of where these cell bodies are for each one. So preganglionic means that these cell bodies are going to be in the CNS in a nuclei. In CNS. But when we get to the postganglionic, these are going to be in PNS, which means they're in ganglia. And so that's why we say either pre or postganglionic. You can name these. For the parasympathetic system, the CNS nuclei is going to be in the brainstem, and it's going to be called the motor nucleus of cranial nerve 10. In the sympathetic system, these are going to be in the interomediolateral gray column. Okay, you can also say IML for that instead of interomediolateral. Both of these preganglionics are going to release neurotransmitter, and they're going to release the same one. So this is going to be acetylcholine. Additionally, both of these are going to have the same receptor types on the input region of the postganglionic. So this is going to be a nicotinic acetylcholine receptor. Remember that when we talked about these nicotinic receptors in the neuromuscular junctions, we said that these are ligand gated. So essentially that ACH is going to bind to these ligand gated channels on the input region. That's going to allow sodium to come in. That EPSP, that positive charge, can set off an action potential that will then travel down the axon and will set off the exocytosis of these neurotransmitters. This is where it's going to be different. For the parasympathetic system, we're going to be releasing acetylcholine again. And in the example we use for this class, we're just showing the heart. So we're going to say that this is ACH, and it's going to work on a pacemaker cell. So it's going to work right here in the pacemaker. But this is going to be a GPCR, not a ligand gated. And it's called a muscarinic receptor. Specifically, this is M2. You don't have to know that. You could just call it a muscarinic receptor, but you can also just call it M2 if you wanted as well. For the sympathetic system, we're not going to release acetylcholine from the postganglionic. Now we're going to release norepinephrine. And that norepinephrine is also going to bind to the pacemaker cells, to a GPCR, but this GPCR is specifically for norepinephrine. It's a beta-1 receptor. And additionally, we can actually also activate contractile cells. So I'm just going to show like they can actually feed to all the contractile cells. So if we look at this now at our ice cream diagram, we remember that this is the brain, like the cortex, I should say, the cortex, it's all brain. This is brain stem. And then this is spinal cord down here in the cone itself. So for our parasympathetic, I'm going to do parasympathetic in orange. We're going to have cell bodies here in the motor nucleus of cranial nerve 10. That's going to travel all the way to the pacemaker cells, synapse on the postganglionic. I should have told you where those cells were. Okay, let's go back and do that. So these cell bodies for the parasympathetic system on the postganglionic, this is going to be in the cardiac ganglion. And then for the sympathetic system, this is going to be in the sympathetic chain ganglion. And so remember, those names are really important. You can look at a test question and you can see the name. And even if you don't immediately remember if it's parasympathetic or sympathetic, at least you can figure out if you're preganglionic or postganglionic. If we're talking about cell bodies that are in a nucleus, it has to be the preganglionic. If it's a ganglion, then it has to be postganglionic. So even if you can't remember the difference between IML and the motor nucleus of cranial nerve 10, if we're just asking you what it's releasing, since they both release acetylcholine from the preganglionic, as long as you recognize it's preganglionic, you're good. Oops, I need orange. That's supposed to be orange. Okay, so this was then the cardiac ganglion. And then we'll do green. We'll do our school colors for our sympathetic system. Let me also label that. That was parasympathetic. This is sympathetic. So this is going to be in the spinal cord, still in the central nervous system, but in the spinal cord, in the IML. This is going to have a really short preganglionic, and it's going to synapse here right outside the spinal cord in the PNS, but in the sympathetic chain ganglion. That will feed, oops, to the pacemaker cell. So the same place that the sympathetic system or the parasympathetic system is feeding, these are going to act on the same pacemaker cells. But additionally, you're going to have branches of this that are going out all over to your contractile cells. So it's going to feed the whole heart. I'm just going to put that in red. You can affect both your pacemaker cells, so you can affect rate and rhythm, but you can also affect contractile cells. And since those are responsible for the contractions of the heart, that means that you can actually control how strongly you contract. We say contractility. This is just pacemaker. So if we were to look at each cell and what's going on, we know that these have to be GPCRs. These are the cells, the target cells on the heart. So you're releasing your transmitter, your neurotransmitter from your postganglionic cells, and it acts on GPCRs. On the pacemaker cell, you're going to have both. What did I do these in? Okay, I'm not sure I'm keeping colors straight, but you're going to have both this GPCR for the sympathetic system. Sorry, that's parasympathetic. And that's specifically going to be that M2 receptor. And you're going to have GPCRs for the sympathetic system. Those are beta 1. So in this case, you're going to be binding norepinephrine. And down here, you're binding ACH. And we remember the GPCR story. We know that we have this carboxyl end on the inside, the intracellular portion, that can bind to an alpha subunit. So we'll do that in purple. We're going to have this boot. I shouldn't draw it across the membrane. It's not transmembrane. A circle and a triangle. That is not how it actually looks, but this is our drawing of an alpha, a beta, and a gamma subunit. And we'll have these throughout the cell, but what's going to be different about them is the alpha subunit. So we can have some alpha subunits that are considered to be stimulatory. We can have alpha subunits that are considered to be inhibitory. And which one a GPCR binds to depends on the structure of the GPCR itself. So the beta 1 receptor for the sympathetic system binds to a G alpha S. Okay, so this is the stimulatory story. Beta 1 cannot bind to a G alpha I. So if you activate the beta 1 receptor by binding norepinephrine to it, it's going to bind to this G alpha S. Likewise here, I'll do this in pink. You've got alpha, beta, and gamma, but this is going to be an alpha I because the M2 receptor can only bind to this G alpha I, which is an inhibitory story. The difference really comes into play when we talk about this adenylyl cyclase. And I forgot to count. 2, 4, 6, 8, 10. Oh, perfect. So this is adenylyl cyclase. It's a transmembrane protein, and its function is to convert ATP, that adenosine triphosphate that we talk about all the time, into CAMP molecules, which is cyclic AMP. Don't worry about the structure of that. Just understand that you can convert ATP to CAMP if it's functioning. So if you have this G alpha S comes over here and binds, this is going to cause an increase in adenylyl cyclase activity. Okay, so if you increase adenylyl cyclase, that means that you're going to be able to make more CAMP, which leads to more pKa. On the other hand, if you bring this G alpha I in, because it's inhibitory, if it binds, it's going to slow adenylyl cyclase down. So you decrease it, adenylyl cyclase, and then you're going to decrease CAMP and pKa. We don't care so much about pKa here in the pacemaker cells. I'm sure it's doing something, but not anything that we talk about in our story. So what we're concerned about mainly is the CAMP, because we have these HCN channels that bind to CAMP. So you need the hyperpolarization, but you also need that CAMP binding. And if you have more CAMP, then you're going to be able to open this channel more. Okay, so this is going to increase your heart rate, because you can have more of those potentials in the same period of time by increasing that ramp. If, on the other hand, you decrease CAMP, you're not going to be able to open the HCN channel as easily. You're going to have less sodium that comes in. And because of that, you'll have longer ramp potentials and a slower heart rate. On the contractile cell, it's just the sympathetic story. We do not have that parasympathetic activity here. So this is just if you have norepinephrine coming in to bind to this beta 1 receptor. Because it's beta 1, not I, that's still going to bind to the alpha S subunit. That's going to increase your adenylyl cyclase. Which is going to increase production of CAMP. And then here's where pKa actually matters. That leads to more activation of pKa. So pKa was protein kinase A. And a kinase just means it phosphorylates. So now you've got more active protein kinase A. It's going to go around the cell and it's going to activate as many things as it can. It's going to put phosphors on anything that can actually take them. Some of the things that you can phosphorylate are things like your DHP channel. Okay, so you had this DHP channel here. You had your RYR channel here. So now you add a phosphor to these, and this is going to increase the activity of these channels, which means you're going to bring in more calcium, which is going to bind to the RYR, which is going to allow more calcium out. And all of that calcium can now go and bind to the troponin Cs and cause the contractions of these contractile cells. And it will hold longer because you've got more calcium. But you've also got a faster heart rate, because if you've stimulated your sympathetic system, it's going to affect both the pacemaker and the contractile. And so now you've got all this calcium that's sitting out here.
My room, so I just took yours.
Okay, so the only way
Sorry.
that you can actually have another heartbeat is to be able to clear this calcium out, reset everything. So the last thing that you can do is affect this ATPase. This is calcium ATPase. So basically what it does is it uses ATP as an energy source to push this calcium back into the SR against its gradient and reset everything. Okay. Just taking a quick peek. Okay, if that's good on heart, unless anyone has any major questions on that. I think Stephanie's been doing a fantastic job answering all of those. Thank you. Okay, we'll go ahead and get.
Do you think, sorry, I was going to say, do you think for the test we'll need to know specifically like GDP versus GTP for the G alpha S subunit or I?
It's possible. I mean, that's something you learned in the last section, so it could be brought up again. So remember that anytime you have an ATPase and it's at rest, it's going to have GDP because if it has, and I said an ATPase, this is a GTPase here, but anytime you have some kind of phosphorase where it's breaking down a triphosphate into a diphosphate, it will do that immediately. So if it's at rest, it's always down to diphosphate. So when this is just sitting here, when these alpha subunits are just sitting here, they're going to have GDP bound to them. When they bind to something, then that's going to kick off the GDP, and now GTP is free to come in.
Thank you.
Of course. Okay, so blood and immune. I know this is probably what most of you really wanted to hear about because we kind of like shoved this into the end. But we're going to start with blood and just kind of go through this quickly. The idea with blood is that you can centrifuge it and spin it. And when you spin it really fast, things with higher density are going to go to the bottom. And so you can actually see 3 different sections of your blood. This bottom section here is called hematocrit. It's really just your hematocrit, your erythrocytes. Those are always going to go to the bottom because erythrocytes are just bags of hemoglobin, and those hemoglobin have iron molecules in them. So that iron is super heavy. So it's the iron that makes it heavy. The second section here is going to be cells that just don't have iron in them. So they're big, they're cells, they have plenty of machinery and proteins and everything else in them, but without the iron, they're just going to be on top of the hematocrit. So this is what we call buffy coat. That's made of all your other blood cells except your erythrocytes. So this is all white blood cells. Or leukocytes, which is the other word for white blood cells. And then finally, most of your blood is actually made up of plasma. Plasma is water, ions, proteins, and then things that aren't actually cells. So you can have platelets in here because they're just a portion of a cell. They're going to go to the top. They're not going to be quite as dense. All kinds of proteins, including secreted antibodies. Okay, so this is where you would have secreted antibodies. If you're talking about a transmembrane antibody that's going to be attached to a cell because it's a transmembrane protein, you'd find that in the buffy coat with the white blood cells. It wouldn't be in the hematocrit because erythrocytes don't have those transmembrane proteins. Okay, so just to bring back bone again, this is the epiphysis of a bone long bone specifically. I just draw a portion of it. And we know that in this portion, we have the spongy bone or trabecular bone. So this was kind of like a sponge. It really looks like a sponge if you look at it. So it has all these cavities in it, which makes it perfect for this red bone marrow. And this is the source of all of your blood cells. So even though we call it red bone marrow, it's both your, oh no. There, we're back. Okay. It's both your erythrocytes and your leukocytes. Okay, so everything is coming from the bone marrow. Don't forget that. I know we talk about thymus maturing T cells maturing in the thymus. We talk about things being in the germinal region. We're going to talk about how we actually make the erythrocytes, but always remember they're always starting in the red bone marrow and move from there. So within that red bone marrow, you have hematopoietic stem cells. And I know that's a mouthful. If it were up to me, if I were writing this test, I would never put this on a fill in the blank because I wouldn't want to make you spell it. But again, don't worry about spelling. We'll give you points as long as it's, what's the word for it? Phonetic. So as long as we can recognize that you're trying to say hematopoietic, that's going to be fine. But hematopoietic, these are the precursors of all blood cells. The only ones we're really going to walk through are going to be erythrocytes. So we'll talk about these erythrocyte pathway. But we can also make our leukocytes, our white blood cells. And then our, what do we want to do? Blue? We'll do blue. Megakaryocytes. Megakaryocytes. There's another one that's fun to spell. So again, we didn't really get into detail on the megakaryocytes. I would just know that this is going to create those platelets. So basically little pieces of those megakaryocytes are going to break off into platelets, and these are going to be really important for blood clotting. We're not going to get into all the cell divisions of leukocytes. You will be expected to know granulocytes, the 3 different types of cells that we talked about that act in the innate system. And of course, you'll have to know your B and T cells, but just know that they come from hematopoietic stem cells. We're not going to walk through that. What we are going to walk through is erythrocytes and their whole process. And I did it again. I really did not leave myself enough room. Okay, so you're going to start in this pathway with a proerythrocyte. Let me make that, what's pro? Green's pro. Proerythrocyte. Okay, so that's your very first, after your stem cell, once it moves down into this path to become an erythrocyte, the first one is a proerythrocyte. And the important thing to remember for this is that it has an erythropoietin also known as EPO, which you're free to use, receptor. Okay, so that's going to be the signal to this proerythrocyte to divide and make more of itself so that you can move on down the line and mature. So this will now go to a normoblast. And normoblast is, I always say I remember normoblast because it's abinormal, because this cell is going to eject its nucleus. So this is the only cell we have in our body that does this. And then it's no longer a cell because it doesn't have a nucleus, which is why we call them erythrocytes and not red blood cells. Once it does this, it can mature into a reticulocyte. So these are named because they're still, even though you've gotten rid of the nucleus and everything else is falling apart, you can still see some fragments of the endoplasmic reticulum. But eventually those are going to break apart. They can't maintain those either. And so from there, it's going to move into the blood. So this was all in the bone marrow. So now you've matured, you're going to move into the blood and mature into an erythrocyte. Okay, please don't ask when it gets full of these hemoglobin because I don't actually know. Somebody asked me that once and I meant to look it up and I forgot. But by the time it gets to be an erythrocyte, it's mature, it's in the blood. This is just full of hemoglobin. That's all it is. It's gotten rid of everything else. It's just hemoglobin. So hemoglobin is this 4 subunit protein. It's going to have 2 subunits that are alpha and 2 subunits that are beta, at least in an adult. And each one of these is going to have iron in the center. And that iron can bind to oxygen. So I'm going to make oxygen green because why not? So now you can bind these oxygen molecules. And this is how you carry oxygen in your blood to deliver it to your tissues. And we'll talk more about hemoglobin and how this is actually functioning when we talk about respiration. So just understand the basics that you've got this 4 subunit. Each one of them has the iron in the center and it binds to the oxygen. For now, we're going to focus more on how we make and destroy our blood cells. So we have from the kidney, I shouldn't do equals. We have cells called pericytes that monitor O2 levels. If O2 is low, it's going to release EPO, erythropoietin. That erythropoietin is a hormone. It's going to go into the blood. It's going to go through the rest of the body, and eventually it's going to get to the red bone marrow. So remember, we had those proerythrocytes in the red bone marrow that had the EPO receptors. So the EPO can bind to those receptors, and that's going to cause this whole system to increase. So now we're going to make more normoblasts, make more reticulocytes, and make more erythrocytes. Now that we have more erythrocytes, we can carry more oxygen, and that should increase our O2 levels. Getting rid of these, we have to do on a regular basis because most erythrocytes last about, I want to say it's like 90 days, 90 to 120, something like that. Erythrocytes, they're short-lived. We'll just say short-lived. And we won't ask you exact date. So one of the reasons for this is the fact that we've gotten rid of everything. There's no nucleus, there's no endoplasmic reticulum. We can't make new proteins for the cell. We can't fix anything that goes wrong with it. So it has a short span. It gets recycled very quickly. And when it gets recycled, that's going to be in the spleen. I should have saved red for the spleen. The spleen is really red because it's lots of blood. So I'm just going to show the spleen, which is an entire organ, like it's just a large cell, because I didn't think to draw this earlier. So the spleen has capillaries in it, just like every other organ would, but these capillaries are kind of special, these splenic capillaries. Because while all capillaries are just a single cell thick, and have some gaps between them to allow ions and fluid to pass through them, these splenic capillaries are what we call fenestrated, meaning they have very big gaps. Big enough that you can actually squeeze these erythrocytes through. So in your spleen, you've got centers all over the thing. I'm just going to show one each, but you're going to have what we call white pulp, which I can't draw white, so I'll draw as yellow. And that's going to be important when we talk about immune. That's going to be a germinal center. But we also have red pulp, which I'll draw as purple. It's already used red. And in the red pulp, in both red pulp and white pulp, actually, we're going to have macrophages. But here in the red pulp, these macrophages that are hanging out, nope. Okay, these are Pac-Man. That's a terrible Pac-Man, but that's a macrophage, big eater, just goes around gobbling things up. So you get these erythrocytes, they get pushed out and they get put into the red pulp, and the macrophage is going to check it out. So these are typically going to have sugars on them because all cells have sugars on them. So those were made before, when it was like a proerythrocyte, before it got rid of all of its machinery, but it can't update them. It can't put new ones out there because it no longer has that machinery. So those sugars will start to degrade. They'll start to break down, they'll oxidize, and the macrophage will recognize that. It can look at it and it can see, are these sugars in good shape, or is this glycosylation pattern now off? And if it's off, then the macrophage goes ahead and eats it and then just recycles all the parts. So if a macrophage is eating this, okay, to recycle this erythrocyte, it's going to recycle the iron because we can use iron. We need a lot of iron, so we want to recycle as much of that as possible. But also, it will get rid of most of the rest of the hemoglobin, including the porphyrin rings. Porphyrin. So that's the structure that was actually holding the iron in place, where the oxygen binds. This stuff, when it is trashed, it becomes things like bilirubin and biliverdin. That's actually going to be, sorry, I'm hungry and my brain is no longer working. It's going to be, the liver takes care of it. It's going to go to the liver. The liver is going to process it. It's processed to the liver, and hopefully you get rid of that. But if for any reason your liver is not working properly and it can't process all this bilirubin and biliverdin, then you'll end up with a backup in your system. And the names, verdin and rubin, suggest that there's some color to it. If it's, not working well and you get that backup, you get what's called jaundice. Because you have this yellowish tinge coming from that buildup. Okay. All right, so lymph. We need to talk about two different types of pressure here, and it's important to remember these because we're going to talk about them again in the next section. We have something called hydrostatic pressure. So when you're pushing blood through the system, that was high pressure. You had to build up all that pressure to get it out of the aorta. And yes, it's going to drop pressure as it moves across the arterioles and into the capillaries. But assuming that this is an artery here, you're still going to have high amounts of pressure from that blood. And that's going to be putting pressure on the walls. That pressure is referred to as hydrostatic pressure. When you get down to a capillary that just has that single cell layer and it has some gaps in there that fluids and ions and things can push out of, that hydrostatic pressure is going to help push all of that across. So now you're going to get things moving out from the capillary into the extracellular space like water and ions and also microbes, pathogens. They're small enough. Some small proteins can also get pushed across, but a lot of proteins are too large, and so they'll stay behind. So you're still going to have like these proteins in your blood. Because you've now pushed a lot of fluid and small things out, you have much bigger solutes left. You've changed your gradient. Now you've actually created a pressure for water to move back. Excuse me. So you would say that these proteins in your blood are pulling fluid back in. And that's what we call the osmotic pressure. So I'm just going to say this is pulling and this is pushing. Both of these are in reference to the capillary itself. So you're either pushing out of the capillary or you're pulling into the capillary. But you will typically have a higher net hydrostatic pressure than osmotic pressure. So even though you're pulling some of the stuff back, you're still going to have quite a bit of it out here in the extracellular space, and it can't stay there. You've only got limited space. And so if you keep pushing water out, you're going to end up with serious edema. So you need to move this out, and we do that through these lymph vessels. So the lymph vessels will pick up the hydrogen, the ions, the microbes, and it's going to move it through the entire system. The lymph vessels are extensive, and they have several lymph node centers. These are germinal regions. These are areas where immune cells congregate. So we don't have anything like that in our blood vessels. So if you get any kind of pathogen into, or an antigen of any sort into your blood vessel, you would never know it unless you can occasionally push a bunch of stuff out, pick it up in the lymph, and then send it through these lymph nodes so that cells in here can check for microbes as the lymph passes through. Eventually, all of this lymph is going to join back up and dump back into the subclavian vein. And so it just goes right back into the vasculature. But this is a nice way to kind of filter stuff from the blood and just check for things and make sure everything's A-okay. We can talk about lymphatic tissues. Further than here. So obviously, lymph nodes were one of them. And we mentioned the white spleen, or the white pulp of the spleen. We also have GALT. So GALT is gut-associated lymphoid tissue. So this is typically in your throat and in your intestines. If it's in your throat, this is going to be called tonsils. And if it's in your intestine, it's Peyer's patches. So your white pulp of your spleen, your lymph nodes, those are actually checking for things that are already internal. They could have been in your blood, they could be in tissues, and if they pass by these germinal regions, then they could be checked out. But we also want to check out the things that we're breathing, the air we're breathing, the food that we're eating, to make sure that none of that is infected as well. So tonsils are going to be checking for things that you're breathing in, and your Peyer's patches are going to be checking for things that may have been in your food. So it's going to be taking stuff that's actually external. I mean, it's like inside your body now, but it's not in your tissues. So it's going to take it across the membrane in M cells. So both of these will move across M cells and into a germinal region. All right. So for the innate immune system, the innate immune system is rapid
and non-specific. So this is the first line of defense. This is, you know, there's a problem. You're not entirely sure what. There's a bug in the system. You haven't identified it, but you know it's there, and you're afraid if it gets any further, it's going to replicate, it's going to create havoc, and you've got to do something about it. So you just throw a grenade at it. Yeah, you're going to damage a whole lot of your equipment in the process, but if you can stop or at least slow down whatever has occurred, whatever's breached your system, then you might have enough time to actually get someone who can identify this and attack it directly to come in and take care of it. So this is really fast, but really messy and chaotic and typically painful. This is when you feel sick. And it's not because whatever pathogen you got is, at least not initially, that pathogen is attacking you and causing problems. It's your own body trying to attack the pathogen that's causing you problems. So it's not actually this simple, but for this class, we're going to break it up into two stories. We're going to say that if we have a viral infection, we'll react to it through this interferon. And if we have bacteria, we react through inflammation. So I would know your SARS-CoV-2 story. It's relevant, and he went into detail, so I would expect to see that. So know that you've got this coronavirus. It has a crown made of spike proteins. Because this is SARS-CoV-2. That's the viral name. And those spike proteins are acting as keys to get into our cells. They can react with receptors called ACE2 receptors. We see a lot of these in the respiratory epithelium, but we actually have ACE2 receptors in a lot of places. In fact, they've discovered some new places they hadn't known about since this virus, because they saw some effects in areas they didn't expect to see them. So these spike proteins can actually bind into these ACE2 receptors and trick it into bringing this whole thing into the cell. So this is going to endocytose. And so now you've got your ACE receptor attached to this virus. And I made it look super big there. Of course, viruses are really tiny. But when it's in this cell now, it's either in the endosome being brought in and it's being sent to the lysosome, or it could actually be in the lysosome. Viruses are just really good at getting out of these containers, these membrane-bound organelles. And in this case, our cells are inadvertently helping. A lot of times the viruses just develop a key and figure out how to get out, but we kind of, screwed ourselves. Or I guess you could say they're tricky enough to have figured out how to use our own proteins. So it's hard to say. I don't know if it's actually on this endosome or if it's just a transmembrane protein. Okay, but this is going to be a protein. I'm going to try to make it look like scissors. That does not look anything like scissors, but okay. That's a TMP RSS2. Transmembrane protein. I forget what the RSS is, and then 2. What this can do is it can actually cleave the spike proteins, which might seem like a good idea. Let's cut this virus up. Maybe it won't be so harmful. The problem is when we do that, we actually expose portions of those spikes that are hydrophobic. And so now those hydrophobic portions can actually interact with the plasma membrane quite nicely, not the plasma membrane, but the vesicular membranes. And it has no problem getting through. So now you've just created a way for this virus to escape out into the extracellular space. It is then going to hijack all of our machinery, make more of itself, and then exocytose and infect all the neighboring cells. So what we do, not just with SARS-CoV-2, but any virus that gets into our system, is when we recognize that there is this infection, we can start releasing, exocytose interferon. Okay, this is a cytokine. It's a signaling mechanism. Okay, so this interferon will diffuse. It's a paracrine factor.
So it's going to diffuse to the local cells and bind to these interferon receptors that all of these other cells are going to have. This is a signal. So the interferon binding becomes a signal to warn these cells that, hey, there's bad guys in the neighborhood, be careful. And those cells are going to lock the door, okay? They're going to reduce the amount of endocytosis so that the bad guys can't get in. They're going to cut down on the amount of exocytosis, because what if the bad guys are already in and they just don't even realize it yet? The least they could do is keep it from getting out and harming others. And then finally, it's going to reduce protein translation so that if it is in the cell, it's less likely to be able to copy itself and then, you know, be able to get out and infect other cells. So these cells, once they're infected, they're goners. We don't have any kind of immune system within the cell that could help kill this. So basically these cells are going to end up being killed to protect others, but they also are very selfless. They will shut themselves down and put themselves in danger in order to try to protect their fellow cells and try to reduce the amount of infection. If, on the other hand, we're talking about a bacterial inflammation, we're going to show the story from a splinter that's coming into your skin. So we have the epidermis. That's just all those skin cells, a lot of which are dead, so we're not too worried about that. But then we had our dermis, and the dermis is made of fibrocytes. Okay, so this was connective tissue. So these cells are not closely connected. There's just a lot of extracellular proteins, this matrix that's in there. So if you have the splinter that comes in and it has this bacteria that's on it, the bacteria is going to start releasing toxins. It'll release cytokines, and cells will recognize that. So these fibrocytes are going to recognize that, hey, there's these toxins, and it's going to cause it to release. I'm not going to show a vesicle here, but of course it's got to be in a vesicle because it's secreted. This is going to release more cytokines. In this case, it's a specific type of cytokine called a chemokine. So cytokine is just a signaling molecule. It could do lots of things, but a chemokine is very specifically a breadcrumb. It basically creates a gradient for other things to follow. So as Walren would say, this is a come hither molecule. It's not just signaling a particular cell, it's saying you need to come to this area. So once you release these chemokines, they can get into the blood and they will travel to different parts of the body. So we're going to say the blood goes this way, and we're just going to say we went from this cell and we're passing all the way through the body, and we're going to the red bone marrow. So these chemokines will now send a signal to the red bone marrow that, hey, we need help over here. There's an infection. And so the first thing it's going to send is neutrophils. So you're going to have neutrophils to the rescue. Those neutrophils are going to follow that gradient all the way back to the site of infection and then be able to move out into the tissue. So neutrophils are one of our granulocytes, which just means it's full of these granules that it can release. And a neutrophil's granules are typically free electrons. These are very oxidative. So these can convert things like water and chlorine and what else can it do? But it's going to make H2O2, which is hydrogen peroxide. And it will make bleach. I should know the formula for it, but it's escaping me at the moment, so I'll just write bleach. Needless to say, hydrogen peroxide and bleach are really toxic. I mean, if you have a cut and you put hydrogen peroxide on it, you can actually see it oxidizing the tissue. Bleach, you don't want to get inside either. You don't want to drink it. You don't want to pour it on an open wound because it will do a lot of tissue damage. So this is where you're going to be creating a lot of collateral damage. You're going to, cause a lot of death amongst your fibrocytes and destroy a lot of the proteins in here. But hopefully in the process, you've also damaged or at least slowed down these bacterial cells. Okay, so normally we would also tell the story of mast cells in this area releasing histamine and the effects that that have, but he didn't have a chance to talk about that. So we're not going to hold you to that. We can't, since you don't know it. But I do just want to say, you don't need to know this, but your histamine from mast cells the effects that these have are going to lead to the 4 cardinal signs of inflammation. And you should know these. These are going to be redness, swelling, heat, and pain. You can also use the Latin terms for this if you like. But just one or the other. You don't have to like memorize both, whatever makes more sense to you. So that's a story we don't tell, but you do need these. You do need the cardinal signs, and that's where they're coming from. So now you've got these neutrophils. They've, set off a bomb. It's like a grenade you set off, and there's just all this destruction. So the next thing that you're going to be bringing in here is going to be your macrophages, your Pac-Man. I'm going to make my Pac-Man orange. So they're not quite Pac-Man yet. They're still immature, and so they're monocytes. But they will also follow this gradient to the tissue and then out. Once this monocyte gets out here, it can turn into Pac-Man. Okay, so this is going to mature into a macrophage. And I shouldn't have made that orange because I made my bacteria orange. Oh well. So this is going to pick up all of that debris, including potentially pieces of the bacteria that got blown up in this whole process. And once it has those pieces, those little antigen fragments, it can actually present those to other people. So it could go back into the blood carrying these fragments that it picked up and show it around and be like, anyone see this? If you see this, let me know. This is a bad guy. He was really bad. We need to mount a defense. So be on the lookout. The other thing that you guys should know in here, and again, we're not going to talk about the why because we're ignoring histamine, but you're going to have endothelial cells, of course, because this is a blood vessel. So it has all this endothelium. And on this endothelium, you're going to have selectins. Now, there aren't typically very many selectins on a cell, on an endothelium, but if it gets a specific signal, which it will get during this inflammation process, it's going to cause it to put more of these selectins on its membrane. And as Walren would say, selectins are not very selective, they're just sticky. So what these do is they basically stick, they're, transmembrane, they're sticking up from the endothelium themselves into the lumen of this vessel. And so as your cells start to come past, you've got your monocytes that were coming by, and you've got the neutrophils that were coming by. Nope, that's gray. There we go. They'll catch. And that helps slow them down so that they can actually squeeze out in this location. So that's how we got them there in the first place. Okay. So we'll move on to adaptive. So once the macrophage has picked up these pieces and it starts showing it around, it's actually becoming part of the adaptive. So the macrophage is unusual because it's part innate. It has this role to play in inflammation where it's just cleaning up debris. But once it cleans up debris and it finds an antigen fragment, it can actually become part of the adaptive because it becomes an antigen presenting cell where it will then show that piece off to other cells. So the adaptive system is slow, but it's very specific. So instead of a grenade, this is a sharpshooter. They're not going to do collateral damage. They're going to identify the target, find exactly what they need to kill the target, and then kill the target. The adaptive system, we're going to talk about a couple of different cells. These are going to be things like your T cells and B cells. So these are lymphocytes. Both of those are considered lymphocytes. And you have to keep them all straight. And I know it seems really tough at the beginning, but I'll teach you a couple of tricks. First of all, when we're talking about these major histocompatibility proteins and the CD8 proteins, the way to remember which one works with which is this rule of 8. So you've got major histocompatibility 1, MHC1, that will always work with CD8 because 1 times 8 is 8. And also 2 will always work with CD4 because 2 times 4 is 8. So that's how you can remember the numbers. Now you just have to remember, what do the numbers mean? And so I remember that MHC1, because it's 1, is the most important one. It's the one that was done first. MHC1 is going to be on all cells. Sometimes we say all nucleated cells, but if it doesn't have a nucleus, it's not technically a cell. So I think you can just say all cells. And basically that also means that this does not include erythrocytes, but this would be epithelium, endothelium, this would be cardiomyocytes, this would be antigen presenting cells. All of the cells in your body will have these MHC1 receptors. So these are not necessarily professionals. These are not necessarily immune cells. So they're not going to be involved in a process of trying to find the right weapon to use against this. They're just trying to see, am I infected? Test me, please, I'm worried. So if they are infected, because you cannot save a cell that has an infection inside of it, the only option is to die. So your MHC1 is going to be on all cells, and your CD8 is a T killer cell. That means your MHC2 is specifically antigen presenting cells, professional immune cells. That's things like your B cells, macrophages. We don't actually talk about them in here, but they're really important. Dendritic cells. And because these are not just checking infections inside, these are professional cells just doing their job, trying to find pieces and then showing them around to everyone else. You don't want to kill them. You don't kill the messenger. You want to actually help them in their job. So these are going to work with the CD4s, which are T helper cells. Okay, so I know that this is what really threw a lot of people. We went through this really fast. I promise it's not as bad as it seemed. And so we're just going to go through this. So you've got 2 cells, and I'm not going to say what cells they are now. I'm just going to mark a few things. This is going to be an MHC receptor. Okay. This is, again, the MHC receptor, but it's in an endosome. So this is an endosome. At some point, you're going to end up with an antigen fragment that gets attached to this endosome. Sorry, gets attached to the MHC receptor in the endosome. That then gets exocytosed. So now you've got your fragment out here being waved around, showing everybody else, hey, look at what I got. Anybody recognize this? Now you're going to have specific cells that can actually check this out. Other professional immune cells, but only certain cells can interact with other cells. And so we have these CD proteins. They're transmembrane proteins. These are always going to be on a T cell. So I'm just going to go ahead and label this a T cell. And those CD proteins will come in and they'll check out the MHC receptor. If it so happens that it's a match, if it's a CD4 to an MHC2, oh, that didn't line up quite right. Here we go. Or if it's a CD8 and MHC1 and this CD protein can bind in here, that will be able to bring the 2 cells close enough together that these T cell receptors, which are very specific, can come in and check out the antigen that's being presented. If this can now bind to that antigen being presented, this is a good fit here, then that's going to activate this T cell to release vesicles that are full of a substance. So it will exocytose those. That's the basic story, and that's true of both your MHC1 CD8 and your MHC2 CD4. So let's look at the differences between them. Okay, we're here again. We're going to say that this is the MHC2 CD4 story. That means that this is an MHC2 receptor, which means this has to be an antigen presenting cell. This could be a dendritic cell, it could be a macrophage. We're going to use an example of a B cell. So this is an immature, naive B cell. And what makes naive B cells unique is that these have transmembrane antibodies. Don't worry about all of the anatomy. Like when we talked about, he got really into depth in it. Here's what you should know about an antibody. An antibody has heavy chain, and then it has light chain. Light chain is just in the Y portion of your antibody. Antibody also has something called a constant region. Basically, that's your foundation. Every antibody is going to have the same constant region, but from there, you can build different variable regions. So this is variable. And it's those variable regions that are actually going to bind to an antigen. And so you make all these different variable regions that will have different shapes and may be able to bind different proteins. So if you have a spike protein on a SARS-CoV-2 virus, there will be a very specific antibody that can bind to a portion of that spike protein. You might actually have antibodies to other portions of SARS-CoV-2. The vaccines were targeted against the spike protein, and they're very effective. Some people who have gotten COVID and did not have a vaccine had long haul, so they still had, they weren't able to get rid of the virus entirely. And it's probably because their antibodies that they created attacked something that wasn't quite as specific and it wasn't quite as effective. Fortunately for a lot of them, once they took the vaccine, which was very specific, it was able to actually clear the remainder of those COVID infections. So this variability is random. We just constantly make new antibodies, you know, like Legos. We just take these pieces and we're like, well, let's try these pieces together and see what happens. And if it happens that you send them out into your system and a pathogen that it can fit with is in your system at that time, then great. That's going to do this whole MHC2 CD4 story. And we'll talk about how then we'll make a whole bunch more of these antibodies because we know they're very useful now. But I mean, theoretically, I could have made a protein that perfectly matches the spike protein on SARS-CoV-2 10 years ago. But because I wasn't exposed to COVID 10 years ago, it wasn't around then, that antibody was useless. And so because it didn't bind to anything, it would actually be trashed. It would be like, oh, that one didn't work. Now you could go through that again. As you make more and more antibodies, you may make that same antibody again later, just randomly. But it's just this process of making all these antibodies and seeing, do any of them work? And so this is time consuming. Theoretically, you could have something that you could come up with one in a day, but it could also take as much as 2 weeks. So these transmembrane antibodies are going to be put out on the surface of these naive B cells, and then these B cells are just going to run around through the entire body and see, can they find something that binds? And if they do, if they pick up this antigen, this could be a pathogen, this could be something bad. So they'll bring it into the cell, they'll endocytosis, whole thing. And then they're going to send it to the lysosome. And that lysosome is going to break that protein down. So we're going to take that big old, sorry, that antigen, we're going to break it into pieces. So now we're going to have, you know, just little pieces of it. So we'll have portions, harmless little portions. It'll then pass those portions off to the sendosome. Where it will bind to the MHC2 receptor here. It will exocytose that. And so now it has this antigen fragment that I can show off to the world. Now, if this happens to be a CD4 protein, meaning that this is a T helper cell, then the CD4 protein will be able to bind to that MHC2. It'll pull it close enough together. And if it so happens that this T cell receptor recognizes that antigen fragment, it's going to stimulate the release of these vesicles, and the molecules that are in this vesicle are called interleukins. These are another type of cytokines, signaling molecules. They will now come over here and bind to an interleukin receptor. And this is going to lead to the replication and maturation of the B cells. It's a process that we call clonal selection. I'm just going to put this over here. Clonal selection is going to make plasma B cells. Okay, so now you know that the particular antibody that this B cell makes is good. It recognizes a bad guy. So you want to make a whole bunch more of it, but you don't necessarily want to have to make all of these transmembranes so that you just have to have a cell that runs around everywhere. If instead you can now make secreted proteins, you can copy those antibodies and then secrete them, then these antibodies can go all over the body and attack and bind to these antigens and signal them for destruction. Additionally, you want to create memory B cells. Now that you've found an effective antibody against an antigen, sure, you beat it back this time, but what if you see it again in the future? You want to keep this on file. So this is going to store the recipe for that particular antibody. And these memory B cells will hang out in germinal regions with a transmembrane. So if it happens to bind to this antigen again, it can immediately just start replicating and creating more plasma B cells that secrete the antibodies. It doesn't have to go through that whole long, tedious process of making random antibodies to see if one works. Okay, and then the second story is your MHC1 CD8. So again, your MHC1 could be any cell. This is any cell within your body. And this, oh no. Let's try that again. Your MHC1 cell could be any cell in your body. So we could just say this is an epithelial cell. This is worried about, is it infected? Did it get a virus that's snuck in there and it's making protein, it's making copies of itself, utilizing all of its resources. Okay, so this is an MHC1. We're not going to be bringing an external antigen into the system. We're going to be testing proteins that are being made in the cell. So this is an internal protein. And we're going to stick that in a lysosome and break it down into pieces. And so we'll have little fragments, and we will push those into the endosome, and that will bind to an MHC1 receptor that's going to get exocytosed. And now if this happens to be a CD8, that means this is a T killer cell. That CD8 will be able to bind, pull this in close enough that the T cell receptor can check out that antigen fragment. And if it recognizes it, 99.9% of the time, it's just your own proteins. It's fine. The T killer cell shouldn't recognize it because it doesn't recognize self proteins. But if it does, then it's going to release perforins. Perforins don't bind to a receptor. Perforins poke holes in cell membranes. So now you're going to have these gaping holes in this membrane. We know we have this membrane for a reason. We're trying to keep a balance on these gradients. We've got all of this calcium, not to mention sodium, that's out here, but it's the calcium that's a big deal. This can now move down its gradient because there's openings, and that calcium is going to trigger apoptosis, which is cell death. So now you've killed this infected cell, and you can go and check all of the neighboring cells and see, did their interferon response actually help and they're clean, or do they also need to be killed? So those are the two adaptive systems that you should know. And I think that's everything I wanted to tell you guys. So if you have any other questions, oh, there's so many chats. Wow, Stephanie, you've been super duper busy. Thank you as well, Emily. Thank you guys so much. Remember, this will be posted so you can go back over. If you have other questions, you can also send us an email. Good luck on your exam, and try not to overdo it. Your brain needs a rest. There we go. I don't want to stop the share. I want to stop the recording.